So in this section, we'll focus purely on the cellular immunity, and in the final section, we'll look at the humoral immunity. So cellular immunity, or cell-mediated immunity, is a form of specific defense in which just the T lymphocytes directly attack and destroy diseased or foreign whole cells, and the immune system remembers the antigens and prevents them from causing disease in the future. Cellular immunity employs four classes of T cells. There are cytotoxic T cells, or T sub C, which we can also call killer T cells, not the same as natural killer cells. These are a separate type of cell because they're T cells. They are the effectors of cellular immunity, and they carry out the actual attack on the enemy cells. There are helper T cells, T sub H. They promote cytotoxic T cell and B cell action, as well as nonspecific resistance. So helper T cells help out a lot of different cells. Then we'll see regulatory T cells, or Tregs. They inhibit multiplication and cytokine secretion by other T cells. Thus, they limit the immune response. They regulate how big the response becomes. And the fourth type of cell are memory T cells. They're descendant from cytotoxic T cells, but they're purely responsible for the memory in the cellular immunity. We'll see how they arise soon. So immunity, both cellular and humoral, occur in three different stages: recognition, attack, or memory. We can also think of them as the three R's: recognize, react, and remember. So in T cell recognition, there are two phases. One is antigen presentation, and second is the T cell activation. In antigen presentation, we first have antigen-presenting cells. Remember, the antigen-presenting cells can be reticular epithelial cells, macrophages, B cells, or dendritic cells. Anyway, once one of these antigen-presenting cells encounters and processes an antigen, it migrates to the nearest lymph node. Here, it displays its antigen to the T cells in that lymph node. When the T cell recognizes this antigen that's being presented by the major histocompatibility complex of an antigen-presenting cell, it will initiate an immune response. There are two classes of MHC proteins. There are MHC1 proteins and MHC2 proteins. The MHC1 proteins occur in every single nucleated cell, so they don't occur in red blood cells because they have no nucleus. They're constantly produced by our cells, and they're transported to the plasma membrane and inserted into it. As they move through the cytoplasm, they might pick up peptides and other either self or foreign particles. When the self antigens are presented, they don't elicit any sort of T cell response because those T cells recognize it as our own labels. However, if viral proteins are presented by the MHC proteins, or abnormal cancer antigens, then we do see a T cell response. In this sense, infected or malignant cells can be destroyed before they do any further harm to the body. MHC2 proteins occur only in antigen-presenting cells, and they display only foreign antigens. So MHC1 proteins could display either self or foreign antigens on their surface, whereas MHC2 proteins will only display foreign stuff. So as they move through the cytoplasm to the membrane, they will only pick up foreign matter, not both foreign and self. So the cytotoxic T cells tend to respond only to MHC1 proteins, and T helper cells will only respond to MHC2 proteins. Thus, our helper T cells will only respond to antigen-presenting cells. This diagram on the right illustrates T cell activation. We'll either have a T helper or T cytotoxic cell that's going to bind to this MHC protein complex. That displays the epitope that specific T cell is programmed to recognize. So the T cells have already been programmed to recognize just a specific epitope. Before there can be any response, the T cell must bind to another APC. In this sense, the T cell has to check twice to see if it really has found a foreign antigen. That prevents the immune system from launching attack in the absence of an enemy. Once we have successful co-stimulation, the T cells will undergo repeated mitosis or clonal selection. This gives rise to a clone of identical T cells, either TH or TC, that are programmed against the same epitope. Some of these cells will become effector cells and carry out the actual attack that we see here, and others will become memory cells, or T sub M. So we have presentation of the antigen by MHC proteins on the cell surface. This will activate either TC or T sub H cells, and you need to have two of them signal that yes, indeed, this is a foreign antigen. After which point we'll have clonal selection, repeated mitosis, creation of many clones of the cells that are programmed against the same specific antigen. Most of those will actually become effectors and carry out the attack on the enemy cells, and some will become just memory cells. So then what's the difference between the T helper cells and the cytotoxic T cells? So these cytotoxic T cells are actually physically attacking the enemy and destroying that cell. The helper T cell will recognize the antigen MHC protein complex and begin secreting interleukins that exert three effects. Remember, the interleukins are chemicals that signal other white blood cells. So they'll attract neutrophils and natural killer cells to the site, as well as stimulating phagocytic activity of the macrophages and inhibiting them from leaving the area. We'll also see stimulation of T and B cell mitosis and maturation. So not only do T helper cells coordinate cellular immunity, but they also coordinate humoral immunity. We can see here that the interleukins will stimulate macrophage activity and leukocyte chemotaxis, drawing things to the site. This is non-specific defense. We'll also see that they secrete interleukins and other cytokines, which will allow clonal selection of B cells. This is humoral immunity. We'll come to that soon. And here, for the cellular immunity, we'll see that interleukins cause clonal selection of cytotoxic T cells, and thus cellular immunity. So in essence, the T helper cells act to communicate and activate all the other means of defense, whereas the cytotoxic T cells just get right to work. So now let's look at how the cytotoxic T cells actually directly attack and kill other cells. When the cytotoxic T cell recognizes the complex of antigen and MHC1 protein on a diseased or foreign cell, it docks on that cell. So let's review. The cell has produced a lot of MHC proteins. The MHC proteins migrate through the cytoplasm to get implanted in the membrane. As they travel through the cytoplasm, they pick up both self-antigens and non-self-antigens if they are MHC1 proteins, or only non-self-antigens if they're MHC2 proteins. After they've done that, through co-stimulation, will cause clonal selection and produce many, many, many more cytotoxic or T helper cells, as well as some memory T cells. So we've addressed how the helper T cells are involved in secretion of interleukins in order to mediate all different levels of defense. Now we're addressing precisely how the cytotoxic T cell kills the enemy cell. So after it docks on the cell, it delivers a lethal hit of toxic chemicals in a similar way that we've seen before. They secrete perforin and granzymes. The perforin ring will punch a hole in the cell and they'll pump granzymes in there. So it kills the cells in the same manner that natural killer cells were killing cells. We'll also see that they secrete interferons. Interferons inhibit viral replication and recruit and activate macrophages that will come along and destroy cells. We'll also see that they produce tumor necrosis factor, which aids in macrophage activation as well as killing cancer cells. So the T cell docks, delivers its lethal hit of this cocktail, and then runs off in search of another enemy cell while the chemicals do their work. Here we can see a cytotoxic T cell attacking a cancer cell. After it injects all of its lethal hit chemicals, the cancer cell will die. And then the macrophages will come along and gobble up the pieces. So now, what goes on with memory? Following the clonal selection, we'll see that some cytotoxic T cells and some T helper cells become memory cells. Memory cells are very long-lived, and they're more numerous than even naive T cells. It takes fewer steps to activate them, so they're able to respond much more rapidly. Upon re-exposure to the same pathogen later in life, the memory cells will launch a very quick attack so that no noticeable illness occurs. Thus, the person is now immune to the disease. This is called the T cell recall response. So obviously, this is an immense amount of material. Personally, I find the immune system material very overwhelming. The most successful way to get through understanding this is going to be to diagram. So spend some time now and put all of this information together in a diagram. Begin with looking at T cell recognition, understanding where the MHC1 proteins are and where the MHC2 proteins are, and what their function is. Their function is to display foreign antigens, or in the case of MHC1, both foreign and self antigens on the cell surface. Then look at the attack role. How do the helper T cells work, and how do the cytotoxic T cells work? And then, how are memory cells created? Once you have all of those components in a diagram, you really have the whole picture laid out. You might use this figure as your basis, but keep it simple. Now we'll be ready to move on and understand how the humoral response works.